So I'm Jason from Roger Kemp's lab at MIT. So today I'm going to talk about microvasculature-on-a-chip model for cancer studies. So today I'm going to talk about two robust strategies. One is about immortalized cells as cell sources. The other is a two-step seeding method that can generate capillary-like perfusable blood vessels and their application for cancer studies. So in all the tissues that we choose, we choose a circulating system, as mentioned by previous speakers, that the nutrients and the cells, they are transported in the circulation system. And particularly, we are focusing on the capillary. Capillary has two features. One is they are perfusable, as you can see from this movie, that the cells or the blood, they can actually transport through the capillary. And another features of the capillary is they have very small diameter. So in some cases, that only single cells they can pass through the capillary with morphology changes. So to engineering this kind of capillary structures, in our lab, we use a microfluidic device. In this device, we actually have three channels. In the center, there is a gel channel. And on the left side and the right side, we have media channels. So in the gel channel, we have microposts that to provide the surface tension to maintain the liquid in the center. So on day 0, we suspend the endothelial cells and stroma cells in a hydrogel precursor liquid, and then we inject it into the in the gel channel. After the gel is cured, we will add the media into the media channel, and then we keep culture those device for 7 days. As you see from this movie, the endothelial cells, they will proliferate, migrate, and make connections. Eventually, they will form something like networks. So they interact with each other. We call it microvascular networks. So as shown here, the endothelial cells are labeled in green. As I mentioned, that the capillary, they have one feature, that they are perfusable. But in our in vitro system, how do we detect perfusibility of the networks? So we can add fluorescent dextrin into one media channel. And then if the networks shown here, they are perfusable, then the fluorescent dextrin, they will flow through the microvascular networks and then goes to the right side of the media channel. So if we took the image of those fluorescent signals, you will see an image like this. So all the magenta, those are the fluorescent dextrin, and they flow through the lumen of the microvascular networks. And if you see, if I show you a two-color image, you will see a perfusible network like this. So the endothelial cells are shown in green to demonstrate the morphology, and the dextrin actually to show you they are perfusible. We can also perfuse cells or beads into our systems. But to engineering such microvascular networks, we're actually facing several challenges. So the first challenge I'm going to talk about is using the batch-batch variations to use in the primary cells. So not only in our lab, but also other labs, we prefer to use the primary cells to engineering those microvasculatures. So in this lab-on-chip model from Roy's lab, they actually developed a very cool vascularized lung on a chip model. But today, I really want to introduce one piece of their data that they actually compare 5 different log number of the human lung fibroblast. They paired with the same type of the endothelial cells, which is HUVEC, human umbilical vein endothelial cells, to generate the vasculature. As you can see, in terms of morphology and also the perfusibility, they are very dramatically different. So this really tells us the variations between the primary cells, they are very dramatic. The other thing is, even though you find a good pair of the stroma cells and endothelial cells, eventually you are going to use all of them because they are primary cells, you cannot passage them a lot, so they have limited lifespan. So to solve this problem, we actually took advantage of the immortalization technique. So by overexpressing H3, human telomerase transcriptase into endothelial cells and stroma cells. So this is actually a Nobel Prize several years ago. That people found that in the primary cells, the telomerase, when the primary cells proliferate, the telomerase will be shortened, and eventually they become senescent. But if we can overexpress this H-TER that they have a telomerase, they can keep this telomerase, so they will become immortalized. So you can think about the immortalized cells are good cell line. So what I found, using the immortalized HUY and the immortalized lung fibroblast, they can form the microvascular network similar to the primary cells in terms of their morphology, such as vessel area, junction density, and also the average vessel length. So this is good. So it tells me at least immortalized those cells, they didn't change the features of the endothelial cell and fibroblast. Since they are immortalized, they can be passaged many times. So I do this experiment. I passage those endothelial cells over 20 passages and also the fibroblasts over 20 passages. But they cannot form good networks at all. But using a later passage of endothelial cells paired with an earlier passage of immortalized fibroblast, they can form good perfusable networks. So this tells me something must be wrong with the later passage of the fibroblast. So what happened? After screening several surface markers of the fibroblast, I found that in the earlier passage fibroblasts, they actually have two subpopulations in terms of CEL1 expression level. CEL1 is one of the fibroblast surface marker. So, but in the later passage of the immortalized fibroblast, they actually lost this subpopulation. So I further sorted out these two populations and formed microvascular networks. I found only the CEL1 positive fibroblasts, they can support the microvascular network formation, but not the CEL1 negative ones. So what's the difference? What's the mechanism? So based on previous studies from our first speaker, Professor Hughes' work, that I actually run a few genes, expression level, and then find CEL1 positive fibroblasts actually secrete all those factors more than the CEL1 negative fibroblasts. All those factors, they are required for vasculogenesis in vitro in the fibrin gel. So this is the mechanism. But since the purpose is to establish an immortalized cell line that we can use, how can we maintain those CEL1 positive fibroblasts? After trying a few conditions, we found that using a serum-free media with a gelatin coating surface, we can actually maintain those CEL1 positive immortalized fibroblasts. And eventually we use those immortalized vascular cells over 20 passages. They are good for microvascular network formation. So this solved the cell sources problem. Well, another challenge that I'm going to introduce you is not only from our lab, but also from other labs. In their previous publications, most of the perfusable networks, they tend to have a larger diameter compared to the ones in vivo. As shown here on the right, these are the mouse brain vasculature. They are, although the diameter is very small, but they are still perfusable. As shown here, they perfuse the dye in cyan color. So the question or the challenge for us is how can we engineer blood vessels that has very tiny diameter, but they are perfusable. So from both our previous and the others' work that we know, by manipulate endothelial cells and fibroblast concentration, we can actually get different morphology of the microvascular networks. For example, if you use a higher density of endothelial cells, but lower density of the fibroblast, we will get a very large diameter vessels, and they are perfusable. As shown here, you see the magenta color, they are detection. But if we use a lower density of endothelial cells with a higher concentration of the stroma cells, then we will get a very tiny vessels. But they are not perfusable at all. It's just because for those larger diameter vessels, they have formed these openings connecting to the media channel, so the dextrin can flow through. But for the very narrow vessels, they lost those openings. All those vessels that are closed. So then, if we combine these two populations, these two conditions, then to have a larger diameter openings connecting to a smaller vessels in the center, probably will solve the problem to get to smaller vessels in the center, but they are perfusable. So to do that, I developed this two-step seeding method. So this is our microfluidic device. First, I seed higher density of endothelial cells into the gel channel, and then I aspirate it from the outlet from here. Because of the surface tension, the high-density endothelial cell liquid, they will be maintained in these regions between microposts. And next, I seed the endothelial cells with the fibroblast into the gel channel. So in this way, in the center endothelial cells and fibroblast combination, they will have a narrow microvessels, but at the outer regions here, they will form a larger opening vessels. So next, I'm going to show you how this two-step network looks like. So in this particular experiment, I actually used the endothelial cells expressing GLP, put them in the center, but at the outer layer, I used the endothelial cells expressing BLP to separate these two regions of the endothelial cells. As you can see, they can form a very larger openings at the region between the microposts, but in the center, they form very narrow vessels, and even very tiny vessels, as I pointed here using the arrows, they are also perfusable. I know this is very difficult for people, especially for the people who sit in the back to see those tiny ones, so I do provide one striking image here to show you that they can form, this two-step method can form a very large diameters at the regions between microposts, and then they are connecting to a very narrow vessels in the center. I also want to show you how robust this method is. So sorry about these crowded images. So this is actually a one-time experiment that I seed 9 devices, and each image is the entire device image. And all of those 9 devices, they are all perfusable. They form a larger diameter openings at the side, and in the center, they have a smaller diameters. Just give you the impression. This is a two-step seeding method. It's really a robust one. And this two-step seeding method can also be used not only for the immortalized cells, it can also be used for the patient-derived ceroid fibroblast. They can be used for dermal endothelial cell dermal fibroblast to mimic the skin model. And it can also be used for brain endothelial cells, parasite, and the astrocytes to mimic the BBB system. And it can also be paired with the iPS-derived endothelial cells. So next, I'm going to talk about the applications of using our model. So because of the time sake, for the time sake, I'm going to tell you 2 examples. One is about the circular, we can perfuse the cancer cells or cancer clusters into our system. So people have found that in cancer patients, unfortunately, they have circulating tumor cells derived from the primary cell side into their circulation system. And sometimes they found that they have this circulating tumor clusters. Those circulating tumor cells, they will go through the capillaries. They will be physically trapped by the capillary because of the smaller diameter. And then they will extravasate it from the capillary and dominate into a second tumor. We call this metastasis. So our model can actually mimic this process by perfusing the cancer cells. But if we have a larger diameter perfusable vessels, as I showed you earlier, then the tumor cells, which show in red, they can stay in the microvasculature, but mainly through the adhesion. This is not what happened in vivo. But using our two-step seeding method, that we can generate very small diameter vessels, then even we perfuse a single cell tumor into our vessels, as is shown here in the right, they will be physically trapped by those vessels. So this is just one example to show you that indeed this two-step seeding method can actually mimic the one what happened in vivo. Another example is we can perfuse tumor clusters. Showing here in red, there are 2 to 4 tumor cells into in one tumor cluster. We perfuse it, and then by using a time-lapse video, we can actually see how those tumor cells extravasate and then proliferate. And eventually, they can dominate one side and then become a smaller tumor. Another application that I want to show you today is about using our model for the CAR T cell killing assay. So as we know that for the CAR T with chimeric antigen receptor T cells, they are very promising immune therapy for the blood cancers, especially for the leukemia. And then But they have they have their limitations for to treat the solid tumor. But our model actually can be used for evaluate the CAR T cells killing efficiency. So in this particular experiment that I co-seed the tumor spheres into this device using this two-step seeding method. So we generate perfusable, very narrow vessels, like as I demonstrated here. Because of magenta, they are the dextrin, they are perfusable. And after 7 days, they form this perfusable network surrounded, the vascularized tumor surrounded by the vessels. We can perfuse the CAR T cells into our device. So this is 24 hours later. The white color, those are either the control T cells or the CAR T cells. We can detect how the T cells interact with the tumor cells. And then we can keep culturing them over more than 72 hours. And eventually we can stand in the dead cells, and then we can evaluate the killing efficiency of this particular CAR T and construct. And also we can collect the supernatant condition medium from the device, and then we can see how those T cells secrete those cytokines to evaluate the T cell response. And so in summary, today I talked about we established unimmortalized cells as stable cell sources, so they are very good for engineering microvasculature. And the other part is we developed this two-step seeding method that we can generate capillary-like microvessels, but they are perfusable by connecting the smaller vessels with larger openings. And the applications for cancer research, for example, we can perfuse the tumor cells or use it for the solid tumor vascularization experiments. So with that, I want to thank my supervisor, Roger Kam, and also David Barbie at Dana-Farber, and also all my lab mates, especially for Shun, Yogurus, Sarah, and Alan. They have their own poster or talks, so please check their works as well. And also shout out to Joe, who is attending our meeting through Zoom. So hopefully he is still here. And also our funding and also our collaborators. And if you really want to know more or use those immortalized cells and know more about the two-step seeding method and other cancer-related applications using our models, feel free to contact me. With that, I'm happy to take any questions. Thank you.